Hey, Adam.
Hi there, how are you?
All right, feels a bit lonely when you're on your own.
I'm glad you joined me. I wasn't sure how this was going to work out, basically.
Same.
I thought I'd get it early.
Just in case, indeed.
Just in case. I was trying to work out how do you share slides as well?
Correct, I have no idea. So I was just looking at the bottom here and seeing what happens.
Yeah,
there's a screen. Where are you based? Can tell by the accent you're probably not in Canada.
I'm in actually Winchester at the moment in the UK.
Okay.
Near Southampton.
Right.
How about yourself?
I'm just up the road from you in Basingstoke.
Oh, okay. Okay, that's where I grew up.
Oh, was it? Okay. That's where we moved back to. So we've been abroad a lot and we came back in 2019. So we ended up here because we had a property here. So we moved into it. We had the property whilst we were away. So yeah, so Winchester's a nice place.
Yeah, hence when I moved moved around myself and then lived in London for about 20 plus years. And then when I was coming out with a young family, the question was, do I live in closer to Basingstoke, which is a great commuting place?
Yeah, it is.
Or do I push a bit further? And my decision was push a bit further.
Smart move. Smart move.
I just couldn't imagine going back.
No,
I think that was it.
No, I'm with you. I'm with you on that. I'm with you on that. So where were you when you were traveling, when you were away?
Well, mainly just in the UK, or mostly. And I did my studies down in Cardiff, and we're based out of Swansea.
Right.
As the team. So a bit down there, and then up to London, and a bit in New York, and then back to London.
Okay. Is CanSense, is that still based out of Swansea, or?
That's correct. Yeah, it's a spin out of Swansea University. So we're just getting going out of there. So we still got the lab in Swansea University. Yeah.
Okay.
Hi, Simon. How are you doing?
Hi there.
Hey. Good afternoon, morning, wherever you are. It's difficult to know these days. If you see
I'm in Toronto, so I don't know where you are.
So it's definitely just over afternoon then for you. Excellent.
Exactly.
Okay. I just wanted to check in with you guys. Everything okay on your end? Any difficulties so far with the platform?
Share screen. How do we do that?
So if you see below, there is a camera and then mic, and then there is third one of shared screen. Do you see that option? So so you can try
that. It's got a red line through it. That's all. Yeah,
the red line. So we can try it right now if you want, just to be double sure.
Do you want to do it first, Vern, and then I'll do it after?
I'll do it. Yeah, no worries. That one. Share. You see that? Magic.
Yeah, I can see it.
Yeah, perfect. Okay, cool. I'll switch that off. Thank you.
And can you share a portion of your screen, or is it all the screen?
I think for the portion of the screen, let me.
No, there's application. Would you like to record this? There's an application window. Okay, fine. So then there is an application tab.
If I'm not wrong, you won't be able to share portion of your screen if it's not an application window. So if it's anything on like PowerPoint slides, nothing that is not on Google Chrome, then you might have some difficulties to share.
Right.
Yeah.
So that's why I didn't share then. Shall I just try that again then? Because that's...
Sure.
Okay, so that's a problem.
And
So yeah, it won't let me share.
maybe try sharing the full screen.
Yeah, I can try that if I cancel everything down, I guess.
Yeah.
Yeah. Sorry, let me just. I'm just wondering how that's gonna. So I need to share the whole screen.
Basically, you won't be able to see anybody, but they'll be able to see your slides. That's what happens. At least that's what does with me.
Okay, so that won't let me do that either.
Okay.
So unless, did you maximize the? Mine's on a PowerPoint, so.
Okay, so that might be an issue. It shouldn't be, but that might be an issue. Okay.
Oh, there you go.
You can see Carola's screen.
Maybe let's just try this one. And
in the meantime, we have 12 people who are viewing this. Hi, everyone. We'll be starting the room pretty soon. We're just finishing off the panel. We'll be having our moderator and our MC of the room coming up here. If you have anything, just introduce yourself in the chat and we'll start this room soon. And guys, you can see how many people are seeing. You can see there's this eye on top.
Yeah,
so there are like
14.
13 people
Yeah.
kind of ghosting on the screen and we can't know who these people are.
It's okay. If you go on the right-hand side of the chat, you can see the people.
Yeah. And you can also put anything on the chat, ask anything to them.
Unfortunately, I'm having a real problem. It's not letting me share the screen at all.
Adam, we can try something. So we have one person who will be there from Montreal Newtech side. How about you share your PowerPoint slide with me? Let me try from my side and maybe someone else can share it for you.
How do I get it to you? Sorry.
Email.
PowerPoint slide. I am writing my email address in the chat.
Okay. Yeah, it's not getting it up there. Okay, I'll email it you. That's rather than.
Okay.
Adam, do you have a very loud clock next to your mic?
No. Is there some background noise there?
It's a very loud clock. That's all. Maybe not you, that's fine. But as you've got your mic open, I didn't know if it was. That's all.
No. So let me just try and just. I've sent over the PowerPoint slide.
Okay.
So you might get that soon. I was just going to try again. I've used Hopin before and it worked absolutely fine.
Okay. Maybe, Adam, if that's the case, let's try this. Maybe you can exit and enter the room again, and we can try again with the resharing of screen, or maybe just refresh the screen.
Or refresh the screen?
Yeah.
Okay.
And Vern, where are you from?
I'm based in the UK. We're literally 50 kilometers apart, myself and Adam, and it takes a meeting in Montreal to get to know that. So there you go.
Hi, Carolina.
Hi, how are you?
I'm good. We're just testing for now. Oh, she left. Did you get a chance to attend the panel so far?
I'm back.
You're back. Okay, let's try again.
Nothing. That was the full screen application window. It worked on both.
Maybe try the full screen one.
Yeah, I did. Yeah. Okay. It's not working, unfortunately.
No. So I have your PowerPoint slide. Let me try from my computer and see. And then maybe we can invite Carolina on the screen and she can do the screen sharing for you.
That'd be great.
Okay. Editing. Read only. Slideshow. Okay. Hi, Mauricio. Can you guys see the screen?
Yes, I can, yeah.
Okay, awesome. So it works for my screen. Okay, I'm gonna come back. Carolina, if you're here, can you please come back on the screen?
Okay.
Hi, Carolina.
Hi,
how
hello.
are you?
Good, thank you.
It's okay. You don't have to, if you don't want, you don't have to turn your camera on. So maybe let's just try if your sharing of screen works. And I'm going to send you a PPT from your email. Maybe just share the screen for Adam. And on his command, you can just move the screen up.
Okay.
So if you see three options, there is mic, and then there is this share screen one with the red line. Just click on that and click on share the full screen.
Okay.
We have 19 people on this breakout session. Hi, everyone. I don't know who you guys are. We'll be starting this breakout session pretty soon. In the meantime, if you can just write on the chat and say hi to our panelists and to me, and we'll start the breakout session soon. Just tiny difficulties, technical difficulties.
Okay, Simran. So do I have, I share my screen, right?
I can't see it right now. Try it again.
Let me see. Okay.
Yeah, it works now.
Yeah.
Awesome. So Carolina, on your Montreal Newtech email, I'm gonna email the PowerPoint slide. Adam, it's only your PPT, right? That's the only one that I should send her, or should I just forward the complete email?
Just the email that I sent you is fine. With the PowerPoint on, will be fine.
Okay.
I don't think there's anything else on that email anyway, is there?
There was some pictures, but maybe it's just like the.
Oh no, it's just the PowerPoint is fine.
Okay. Carolina, check your email if you got it. Hi everyone on the back end. Again, can you please say hi to us? We feel lonely. Hi Valerie. Hi Mauricio. Hi Carla. Hi Olivier.
Hello. Hi everybody.
Finally, you're here.
So
hi Samantha.
Yes, Hassan will join us. He just leave the main panel, and you have to log and hop in, and he will join us and Alejandro too. So excellent.
Hi everyone.
We're missing Carola from AltisLabs.
Hello, I'm here.
She's already excellent. Everybody's on time. Good.
Olivier, we just had a difficulty of sharing Adam's screen. So Carolina is gonna do the screen. Hi, Hassan.
Hi, everyone.
You finally made it.
Yes, finally.
How was your panel?
So it was great.
I tried again to see it, but again, as usual, I was handling the technical part, so I couldn't. Hi, Alejandro.
Hello, everybody. Thanks for your patience. Hassan, I hope that you enjoyed the conversation with me.
It was great.
Awesome. Thank you very much. And thank you, everybody, for joining in this room. Feel welcome. This is your home. And we are going to have a nice discussion here with everybody around the table. You already know, like, very much who the others are because we've been talking before. But so Montreal Newtech is here to foster the collaboration between the startups, researchers, and industry. This great coalition, the objective is for companies such as Servier, scout for new technology, and for the people like you, researchers and startups, to be able to showcase your technology to the companies, and also like brainstorm with them and look for potential solutions. Montreal Newtech has roles such as super connector, like someone like Simran, like myself. Our role is to get to know each one of you, your needs, your what you're looking for, and then the same for everybody around the ecosystem, and then try to connect the dots and just to connect them with each other. So without further ado, I want to pass the word to Olivier Thomas, who is from Centech, and he will be our amazing moderator for today.
Yes, thank you very much, Alejandro. So welcome everyone to this breakout session. I'm really happy to leading the discussion today. So my name is Olivier Thomas. I'm at the lead of the Collision Lab. Collision Lab is the open innovation hub of Centech, a university incubator specializing in deep tech in Montreal. So I will present all the participants in our session. So we have Hassan Chouaib, is head of AI Hub here at Servier and Data Science Ambassador. So welcome, Hassan. We have Carola Schmidt, Director of Medical Imaging of AltisLabs. Welcome, Carola. We have Casandra Mangroo. Well, now it's Carolina from BenchSci. No, Casandra is here. Okay, she's just right on time. So Casandra, welcome.
Hello, everyone.
Casandra, VP of Science of BenchSci. Adam Bryant, CEO of CanSense from UK. So thank you very much from the other side of the ocean. And we have Vern, Biasi, Chief Partnership Officer at Cyclica. And I think Vern is in UK too. So welcome. So it's an international panel today. So we will have a discussion. We have around 55 minutes for the discussion. So we have time to have a very interesting chat together. So we'll start with the reverse pitch from Hassan. We will explain what Servier is looking for. And then after that, we will have the 4 startups. So it's really easy. I take the alphabetical order for the startups. So we will start with AltisLabs and BenchSci and then CanSense, and we will finish with Cyclica. And so Hassan, you have around 4 to 5 minutes to present your reverse pitch. And for the startups, you have 2 to 3 minutes to go. And after that, we will start the discussion. So Hassan, the floor is yours.
Yeah, I will try to share my screen. So you can tell me if you see my screen. Okay, so do you see my full screen?
Yeah, excellent.
Okay, good. So today we'll try to discuss how could machine learning and multimodal data can improve the data patient selection and prediction of treatment of response for personalized medicine. So some words about me and Servier. So my name is Hassan Chouaib. I'm in charge of the Artificial Intelligence Hub of Servier in Montreal, Canada. It's a new, freshly new hub launched 4 months ago. So Servier is an international pharmaceutical group governed by a nonprofit organization with presence in 150 countries with 22,000 employees. So today we have medicines in 5 therapeutic areas with a main focus on oncology. And today we have 23 to 25% of our brand name revenue reinvested in R&D, with 50% of this 25% invested in with focus on oncology, with 14 projects in clinical clinical development and 22 research projects. So why oncology today? We know that cancer is a major cause of death worldwide, and the success of cancer therapeutic in clinical trials is the lowest of all major disease, and less than 5% of clinical trials get the regulatory approval. And but we still know that developing therapy in oncology is still very, very challenging, especially with the large variability in response of treatment, not just across the different type of cancer, but even across patients for a same type of cancer. And it becomes even more challenging when we are working with a combination of treatment. So that's why we believe that the predictability of treatment of response could improve the patient care by identifying the right therapy at the right dose for the right patients. And with all the data that you have for the patient, all those data can be used to predict all the treatment response of in oncology. So what kind of data? We have different type of data. We have omics data and non-omics data. All those data can be used to extract, to help to gain insight in the mechanism of disease progression and response of treatment. But more we add data, more we add the complexity to the problem. For sure, when we talk about data, we're gonna talk automatically about artificial intelligence. So today, artificial intelligence and data can help to provide powerful tools on the identification of new markers of response, and to define more relevant selection criteria of patients. But today, all the researcher, we have a lot of research paper around this subject, and some existing AI solution. But we still have all the AI applications that we have today in oncology are not mature enough, with a lot of challenges at different level on data collection, data analysis, data integration, and modeling. So we know that we have a lot of data coming from different sources with different type and nature, structured data, non-structured data, qualitative, quantitative, and to normalize and to pre-process all those data is still a big challenge in this domain. So in this at this level, we need the business knowledge too, the domain expertise knowledge, and it's sometimes a challenge to have multidisciplinary teams to accelerate the development of this kind of solution. The interpretability and the explainability of AI models is still very, very important, and it represents a limitation today. And this kind of problem can be challenged by traditional mathematical models like pharmacometrics. And finally, a big challenge today is the lab-to-lab variation that can limit the benefit of collaboration and initiative in the industry to combine data and results obtained in several studies. So, and finally, so we have teams working on this kind of challenges, but we still face challenges on the different levels that I mentioned before. And just to finish, to mention that Servier invests $1 billion every year on the R&D, excluding acquisition and partnership. We have 40 research collaborations, and we are open for new collaboration on this kind of challenges and subject. Thank you very much.
So thank you very much, Hassan. So before we start with the startup, I will remind, as Alejandro write in the chat, if you have questions for the people all around the table, you can write them in the chat box, and we will push that question to our participants. So now we'll start with Carola from AltisLabs. So Carola, it's your turn. So you have up to 3 minutes. Excellent. We will see your screen.
You're on mute.
The micro button, yeah.
I got it, sorry.
Okay.
Okay. Hello, everyone. Thank you for joining today. My name is Carola Schmidt. I'm the Director of Medical Imaging at AltisLabs, a Toronto-based company that strives to accelerate drug development and clinical decision-making through predictive imaging insights. Circling back to precision medicine, at its core, it's finding the right treatment for the right patient at the right time to yield the absolute best possible outcome. But the way we're currently measuring this outcome and overall treatment success in clinical trials leading towards regulatory approval and later on in clinical practice is flawed. We know there is no tight correlation between those imaging biomarkers like response that are generally being used in oncology trials and the outcome that patients and their treating physicians care most about, which is survival. Several anti-cancer drugs had to be taken off the markets in recent years because they just didn't deliver what they promised to pre-approval. We're in the situation where we have a high rate of late clinical trial failures in combination with vast amount of money spent on R&D, but as Hassan mentioned, only a small fraction of those drugs actually make it to market. Anatomical imaging like CT, the working horse of clinical trials, contains so much information that's just not being used and included in assessments. We know, for example, that overall body composition, peritumoral tissue, or cardiovascular comorbidities significantly contribute to a prognosis of a patient. At AltisLabs, we use deep learning to extract all this information from body volume and kind of put together the puzzle pieces to really create a personalized prognosis for each patient. As an example, we were able to predict reliably 2-year survival in a cohort of NSCLC patients and really stratify those patients according to their overall risk profile. We offer an end-to-end solution for all clinical trial imaging assessment related tasks, from timely upload and ingestion of imaging studies, real-time prediction generation, and then this can form the basis for further trial decisions, trial planning. We actually help pharma to gain more insight into their imaging. And some use cases I can see is like, for example, patient stratification, trial enrichment, or an earlier determination of efficacy or a lack thereof. We not only work with paying pharmaceutical companies like Bayer, who guide us in our product development, but also with the largest Canadian cancer centers, who provide us with 15+ years of patient-related data like imaging, labs, outcome, and treatment for our product development and testing. We published or co-published at ASCO, and we're in the process of submitting a manuscript to the Journal of Clinical Oncology. Thank you.
So thank you, Carola. The 3 minutes are over. So thank you very much. And then we will switch with Casandra from BenchSci. So Casandra.
Just going to share my screen.
Excellent.
Can everybody see?
Yeah.
Yeah.
Go ahead.
Oops, 1 second. All right, so my name is Cassandra. I'm the VP of Science here at BenchSci and one of its founding members. I'm very excited to share with you today about what we do and how we aim to bring medicines to patients years faster. Before I hop in, a little bit about us. We're a Toronto-based company. We have about 130 employees. We work with 15 of the top 20 pharmaceutical companies. We've raised $60 million to date and are very grateful to be the first Canadian company that Google has invested in. So what is the problem that we're aiming to solve? Well, we've learned that $150 million is invested in individual assets within the drug discovery pipeline. It takes 5 to 10 years of preclinical research and development to actually bring those assets to clinical trial. Yet 9 out of 10 medicines that actually enter clinical trials fail. Well, why is that? Biology is very complex, as we've heard earlier. Diseases are often not caused by single genes or proteins. It actually takes many experiments, 7,000 to be exact, and 5 to 10 years of work to understand the underlying mechanisms of diseases and bring single treatments to clinical trial. However, we've learned that that preclinical process is actually quite inefficient. Only 20% of the experiments that are done in preclinical R&D are actually needed to advance each medicine. And by eliminating the unnecessary 80% of experiments associated to redundancy, irreproducibility, and the use of inappropriate reagents, we can actually improve the efficiency of preclinical R&D and improve the movement of medicines to patients years faster. So how do we do that? Well, BenchSci has spent the past 5 years developing machine learning technologies that extract information from both text and image, understanding the key experimental entities of the protein target, as well as applications and surrounding experimental context. This information is often buried and not available to scientists. Now, by pulling this information, standardizing it, and putting it through our application, scientists now are more informed in their experimental design and can address key challenges and questions that they face during preclinical R&D. Namely, what are the most impactful experiments to conduct? What are the most efficient ways to perform them? And how do we increase the reliability in informing clinical trials? By doing this, and in partnership with our pharmaceutical companies across many different disciplines, including oncology, we can help move novel medicines to patients 50% faster by 2025. And that's it from me.
Okay, thank you very much, Cassandra. So now we will switch to Adam from CanSense.
Hi there, it's Adam. Someone's going to help with my slides. Carolina, thank you.
Yes. Okay, Adam, I will share my screen. Okay.
That'd be fantastic. Yeah.
Excellent. It's coming.
Okay.
Excellent. So you can go, you can start, Adam.
Can you just go on to the
full screen?
Full screen on that, please. Carolina, could you go full screen on that, please? If that's possible.
Okay, give me 1 second. I put full screen.
No problem. We're in direct, so sometimes it happens.
Perhaps I'll just start anyway.
Yeah. Oh, we lose the screen.
Okay.
Well, that's okay. We can work without it. So we're CanSense. We are focused on the early detection of cancer. And why now? We've been able to combine Raman spectroscopy with AI technology on a simple blood sample, so a liquid biopsy. Our first focus is actually bowel cancer targeting when it is treatable and curable. Next slide, please. We are focused on delivering this into primary care practice in the initial instance and working with pharmaceutical companies as well. With helping with their clinical trials as you will see going forward. We're providing them with a quick and accurate diagnosis, reducing the need for invasive procedures like colonoscopies, while speeding up the bottlenecks for other cancers. We all recognize that we're at the lower end of the league tables globally with this illness, and also the fact that cancer in itself needs to be detected early, and traditional diagnostic methods like colonoscopy services are just under so much pressure, only set to get worse as disease becomes more prevalent, and more prevalent also within the younger age groups. The diagnostic services, traditional ones, just can't cope here in England and across the globe. We need to rethink diagnostics. Next slide, please. I think we're all familiar with the bowel cancer in itself and the problems associated with it in early diagnosis. 60% of people are still presenting at late stages. And why is this? Next slide, please. The rationale is, in the most part, because the way that we are testing people and the pathway is inefficient. And that really leads, because people do not want to send their thesis off in envelopes and they do not want to have a colonoscopy. What we're offering is a simple blood test that people are familiar with great degree of accuracy, high degree of confidence, that can rule out cancers immediately. And what's more exciting, perhaps, is that we're not just providing a reduction of colonoscopies by 50%, so less invasive treatment for patients. We'll also be able to detect, next slide please, precancerous polyps themselves with a high degree of accuracy. And this is where pharmaceutical companies that we've spoken to get really excited, being able to identify patients before the cancer's even formed from changes in their blood. Next slide please. We're doing this by shining effectively laser light at blood, which produces a unique spectral fingerprint for each patient, which then is compared to our AI libraries for bowel cancer, but it could be compared to other AI libraries for other cancers. And the diagnosis is given straight to the clinician. We in the UK, next slide please, are not alone in the healthcare burden that bowel cancer has itself. On globally, it's a 265 million cost saving that we could provide to our NHS, or a 4.2 billion cost saving we could provide globally in just the reduction of colonoscopies, not including late stage diagnosis. And perhaps I'll leave you with this, which is next slide, please. Our application. We mentioned the fact that we could look at symptomatic patients and give a GP a better tool in their toolkit to identify patients sooner and get them treatment quicker that then could be curative. But furthermore, we could look at reoccurrence monitoring within our patient set as well. And we've talked about working with then pharmaceutical companies in their drug discovery, providing really, next slide please, a low cost and affordable solution that can constantly test patients by just doing one simple blood test. And I'll leave it there, please.
Okay, thank you very much, Adam. So now we will finish with Vern from Cyclica. So Vern, so thank you for joining us, Vern, because we just discussed less than 24 hours ago, and you're with us, so that's the magic of.
Yeah. Can you hear me? Can you hear me? Okay.
Yeah, we hear you.
And we see the slides.
Yeah, we see the slides.
Fantastic. Technology works. Okay. Thank you. Thank you, firstly, for the invitation. Glad to be here.
Yeah.
My name is Vern. I lead the partnerships team at Cyclica. And to talk, just to give you a quick picture and overview of Cyclica. Cyclica is a biotech company which has technology at its core, and that technology, developed by ourselves and patented back in 2013, has allowed us to really investigate and understand and appreciate the thesis on polypharmacology. Polypharmacology is about how, when a drug enters the body and does more than just one thing, and that understanding and that thesis has allowed us to build technologies to appreciate and investigate polypharmacology, called Ligand Express and Ligand Design, and they're powered by two deep learning and machine learning engines, Matchmaker and Poem. You can see the rest of the company's details there. Sorry, let me go into presentation mode, it'll be easier. So when a drug enters the body, it exhibits not both on-target, hopefully, effects, but also a number of off-target effects. And those off-target effects can typically derail a drug discovery program as it goes through preclinical and then into the clinic. And our work is looking to try and understand those off-target effects, and also then from there also design molecules with on-target effects and trying to reduce off-target effects. That hypothesis and that technology has allowed us to work on over 100 programs with different size companies in different disease areas and target classes, including obviously oncology. And with the statistics that we've done, we have approximately a 64% success rate of providing actionable hits to our partners, which means those are molecules which we've designed, they've synthesized and tested in their wet labs, and has allowed them to either build chemistry programs on the back of them or use them as tool compounds to investigate their biological hypothesis. How we do that is used a deep learning engine matchmaker and a machine learning tool Poem in integrating on a platform of ligand design, which allows us a proteome-wide approach to design and a multi-objective approach as well. So in the design of molecules, we apply selective pressures in the molecular design, be they molecular features of the molecule, ADMET properties which we can design in, as well as use the targets and anti-targets to provide evolutionary pressure on the molecular design. And through cycles of design and applying these pressures, we generate molecules which have favorable properties for the targets that the partners are interested in. Those molecules then can go into ligand express, where we can then also investigate the unanticipated potential off-target effects that might be occurring, as well as also the modulatory impact that those molecules may have. And just to show a particular case that we've done, in fact, with a researcher at University of Toronto, where we were involved in identifying a selective molecule for an EGFR triple mutant, we, in about 6 to 8 weeks, through some screening and synthesis of molecules which were relevant to the program, identified a compound which generated significant efficacy and selectivity over the wild type, so Cyclica number 7, and that allowed the researcher to pursue and investigate further a new chemotype, which allowed them then to obviously publish, and then we actually continued collaboration, generating even more potent molecules and selective molecules for him. And that is, in summary, Cyclica's offering to this program.
Okay, so thank you very much, Vern. So now we will start the question and the discussion. So Hassan, do you have question first for the startups, or we go for the more generic question? You're on mute, Hassan.
Can start with a question for Carola. Today you did your experimentation on NSCIC, I think, data sets.
Mm-hmm.
And if I remember well, so in the NSCIC data set we have big tumor, and you don't think that having a big tumor and trying to extract features to link with the survival can have some bias, because we have in the data set we have just big tumors. Did you try? Do you have the chance to try on other data sets?
Well, we started with a data set that was a bit heavier on the side of earlier tumor stages. So we were a bit short of later stage patients, stage 4 in particular. So that was a bit, but we were still able, we separated our entire patient collective and created 5 different groups where we kind of spread out all the stages equally. So, and we tried to account for this asymmetry a bit by creating several groups, and those groups were kind of stratified according to their stages and make them kind of symmetric. We're currently working on a new project where we incorporate 15 years of data from the University Health Network here in Toronto, where we have all different kinds of tumor stages, tumor sizes, and outcomes. And I think that'll make our algorithm more robust and than it was just using the initial data.
You're on mute, Hassan. Sorry.
So you are focused more on lung. Do you have any plan to extend your solution on other pathologies?
Yes, absolutely. So we have a colorectal cancer project in our pipeline that we're waiting to kick off. Generally, we're focusing, of course, on areas where medical imaging is used to monitor treatment efficacy, because we're based on imaging. We do have a current project regarding pulmonology, specifically community-acquired pneumonia and prediction of ICU admission, ICU stay length, and also cardiovascular disease, which kind of interlinks with our NSCLC project, because we know that people who tend to develop lung cancer often have other risk factors, including cardiovascular disease. So that would kind of like play into this. So those are different areas we're looking into currently, and then focusing more to extending into the oncology sector.
Okay, thank you so much.
You're welcome.
Thank you.
So maybe I have a question for Vern too. Today, so as I understand, so you have deep learning model to generate molecules, if I understand well. That's correct. Okay. So and you are using another models to predict the ADMET properties and integrate that in new deep learning models to generate molecules. That's it?
Correct.
Okay, good. Can you just tell me a little bit about the platform that you have today and how it can be used in the workflow of modeler and chemist?
Yeah, absolutely. So if we start with Ligand Express, which is our protein-wide platform, so that has the protein structures of about 9,000 human proteins. And in addition to other sort of models that we've built into that. And that allows us to, if you like, understand the interaction of a molecule with the proteome. So which potential proteins is that molecule most likely to interact with? So identifying phenotypic screens would allow us to interrogate molecules and identify what protein targets that could hit, as well as also repurposing, which is something which we've been doing a lot of, obviously, in the recent COVID pandemic. A lot of molecules have come to us to identify if we could hit potential targets. So that has a user interface, and that is something which someone in a laboratory could log into and have access to, okay, and therefore can also use that within their workflow within a company. And we can actually build that API on your facility or off-prem or on-prem for yourselves. It is hosted in the cloud typically for us. From the ligand design perspective, that hasn't got a user interface. And what happens there is the partner comes with the biology of interest, with the targets that they may be interested in doing, and also anti-targets. So the advantage that we have is that we can do multi-target design. So we can design to more than one biological target of interest, as well as away from targets that may not be of interest, or maybe want to be some selectivity. That information is provided to us, obviously. We then search and work with our protein structures that we have in our Ligand Express platform. And then we will design through a series of cycles molecules which will have certain molecular features, so hydrogen bonding, molecular weights, or what type of structures do you wish the molecules to have, as well as then build POEM models with ADMET properties, which do you want it to pass the blood-brain barrier? Do you not want it to pass the blood-brain barrier? Do you want it to have other types of features? And we can apply those pressures to the molecular design, as well as actually use the structures and the features of the protein target to actually apply evolutionary pressure as well. So those three things are applied in sequence, generates a series of molecules. Those are then fragmented and go through the same cycle again and again and again until we get to what we call convergence, which is basically when the output of that is the same as the output of the previous cycle. And that way we can design molecules which seem to be the best with those properties. Those features of ADMET properties or other features that the companies such as Servier, where you have generally a lot of data in large pharma, we can incorporate those into POEM models. So we can build POEM models for yourselves, and those models can then be tailored to the targets that you're interested based on your data. So we can use data from partners to actually build custom models for POEM, but also custom matchmaker models. So we can incorporate information which larger companies have, quite frankly, the smaller ones don't, into designing more bespoke POEM and matchmaker models to suit the targets which partners are looking to interrogate. And those are the two platforms. That last platform, Ligand Design, is not got a user interface, so we have to partner. But the matchmaker and POEM, can be put on your premises if that's something which would be of interest.
Okay, so as you mentioned, so you have already an APIs to interact with the platform. So just to facilitate, yes. For example, for modeler that use, for example, tools like design, etc. So we can interact directly with the platform to be able to get results and integrate with an external tools.
So we are just building at the moment on-prem the system for one of the large pharmaceutical companies. The API will be here in about 3 or 4 months' time, so that you can do exactly what you want. So it's something that we've started to get significant interest in. So it makes sense to build an API to then facilitate that for others.
Thank you so much. So Olivier, I will let you go with your question for now. So after that, maybe I will have questions for others.
Okay, so we still have around 23 minutes because we have to find 5 before 3. So I have a question. Perhaps we can. I will combine the 2 questions in one. So how does your solution help accelerate the development process of therapeutic solution? And perhaps you can just explain in a few words at what drug development stage your solution could help. Are you on the target identification, preclinical, clinical trials, or different stage? So perhaps we can start with Casandra. If
answer the second question first. So we really focus on the preclinical stages. One of the challenges that we hear from many of our scientists is that they often receive, compounds or targets to start testing, but they have no idea how to start that pursuit. So the challenge really is about going through all the disparate sources to understand how to actually interrogate that particular target, understand the underlying mechanism of action, and figure out, what are the most appropriate experimental designs and reagents to actually test them. There have been many instances where an inappropriate reagent was used, and there have been many even published with COVID where an inappropriate reagent was used and it caused delays in the preclinical pipeline that could have been avoided if more effective experiments and resources were used at that stage. And just to share a story, I didn't get a chance because we only had 2 minutes. Just an example of what this is like. We've had groups that are working to do screens to select the appropriate RNAi, antibody, CRISPR, and they're spending weeks to months to find one that's appropriate for their specific experimental context because it doesn't work in every tissue or every disease context. They searched for 8 weeks and then an additional 4 just to find one product. And when they tested our platform, which is a application that's web-based, we did a search, filtered down for their target, the specific cell type that they were working in, and found the one product it took them 12 weeks to find in 30 seconds. So these are just tools that scientists can leverage to make more informed decisions, make better decisions, do more efficient science in the preclinical stages, and shorten the time it takes to get to clinical trial.
Okay, so thank you, Cassandra. And Adam, in your case?
I think ours is clearly slightly different. We're focused on the diagnostic side of things. Yeah. So, I mean, the way that we really just, as I was trying to say in the thing, is slightly different from most people, is that we are focused obviously on that detection side, which means that if there are either drugs that are being used in chemotherapy or people have had operations to remove such cancers, then it's our minimal disease detection that we can do that enables us to get a better understanding of whether drugs are working or not. And at the moment, that's around bowel cancer, but there's no reason that that shouldn't be for other cancers. In fact, we've looked at pancreatic and breast cancer just by analyzing the blood. So where it's useful and where we're speaking to pharma is that, partnering with them on their clinical trials around their drugs and working with them in that way.
Okay, thank you very much. And Carola? You have to unmute.
Yeah. Hi. We're more in the clinical trial phase of things because we're focusing on imaging related tasks. which allows biopharmaceutical companies to gain more insight into the imaging, which is generated anyway, but not utilized. We're currently using biomarkers like RECIST, where we measure one-dimensional measurements and add them up to detect some arbitrary, see if a tumor or a tumor burden exceeds an arbitrary threshold, and then declare it a success or a failure. Whereas we kind of automate this entire process. Not only do we offer to volumetrically capture the entire tumor, because this has shown to be advantageous, because tumors don't just grow in one dimension, but they expand differently. We also enable pharmaceutical companies to detect treatment success earlier and more meaningful endpoints, other than those surrogate biomarkers like response or PFS. We can tell, your patient's survival is changing. They're surviving longer now, which we believe is a lot more meaningful than saying, okay, this tumor has changed in 44, the tumor volume has changed 44%. What does that mean? Does that really kind of influence how long this patient or how well this patient is living? So we're enabling with our platform earlier detection of trial failures, kind of moving resources to more promising indications and projects, and identifying those projects that are really promising and able to really create meaningful data to be used then in the regulatory approval process.
Okay, thank you very much, Carola. And Vern, so you already respond a bit.
Yeah, a little bit. So we're very at the drug discovery front end, so the identification of hits. We then have an ability to do hit expansion. So different scaffolds are available, through the technologies that we have. I think one of the things that we found particularly useful for some partners is that in the design, we can constrain features in the design which are known to be important or actually known not to be wanted. We can make sure that those are not included in the design as well. So it's allowed us to scaffold hop with features that remain important for particular interactions on certain targets. So hit expansion, ability to do scaffold hopping, and then we're also doing, we're now starting to move into, helping to define leads for companies as well. So slowly we're moving the technology forward to get greater impact further down.
Okay, thank you very much. So
Sure.
Hassan or perhaps Alejandro, do we have questions from the attendees? Not for the moment. So for the
it has been like a nice pace of conversation, but so far I haven't seen any question regarding the conversation. There's people that is joining us from the Caribbean, from Latin America, interested in the subject. They just want to learn more about what all of you are working on.
Okay, excellent. So
maybe I have a question for Casandra. Can you tell us a little bit more about the technological stack that you are using now, and especially on AI level?
Yes, of course. So we started with text-based machine learning. So those are NLP deep learning algorithms. We have over 40 different biomedical entity recognition models that are extracting, protein target information, reagent use applications. We started that way and soon realized that wasn't enough. So we actually developed proprietary image or vision ML algorithms. So semantic segmentation models to parse through complex scientific images, detect subpanels, classify images into the appropriate application so we can identify IHCs, graphs, etc. We have text-based extraction and OCR models that actually can extract embedded text. So this is no matter the orientation, horizontal, vertical, diagonal, we realized a lot of information is actually missed if you're only depending on NLP-based algorithms. So by extracting that and leveraging our ontologies, and that goes across bioinformatics, disease, accounting for synonyms, aliases, how all of those things are cited in the various sources, we can then capture that and make sure that it's standardized so that actually makes information more searchable and actually brings those insights forward, regardless of how it was written by the scientist, because we're looking at the experiment objectively. So we're looking at both the text and images, and we can also look at sources if it comes only in text, only in image, and all of that is combined, and we link all of these entities back to the experimental output. So when scientists are interacting with the platform, they can search by their target, cell line, cell type, disease context, even subtypes of cancers, and see the actual experimental evidence that's linked to it so that they can go in and see what reagents were used at that point, what was the experimental design that was leveraged to study that particular cancer, and then use that information to inform their own experimental design. And we found instances where scientists didn't even know certain experiments existed. And this is something that's not just applied to publications, it's to their own internal data. We take data from core facilities, third parties like Human Protein Atlas, and bring all of that together so scientists can see it all in one place.
So it's a web-based application?
It's a web-based application at the moment, but of course, similar to what Vern mentioned, there are also bespoke offerings that we have where we can apply that machine learning technology to internal data to extract insights from that as well. The redundancy that we see within organizations is very high. We've seen scientists performing the same experiments across different labs, not knowing those experiments were happening down the hall. And that's just because they have no visibility on any of the data that's happening internally. And it's actually quite surprising how prevalent that is. And by bringing to light the internal insights as well as overlaying that with external insights, we're able to really bring it all together. So,
I have a question.
So much.
There's finally somebody here, Olivier. Anthony Canavici, I hope I pronounced it well, is asking, and I'm guessing this is a question for Casandra. What is the error rate of these image-based ML algorithms? For example, false negative or false positive reads?
Yes, that's a great question. Obviously, your algorithms are only as good as their training set. So one of the things we learned is that you can't use anything off the shelf. We actually train the algorithms ourselves. And the data before it goes live on the platform is actually pre-curated. So we measure for our algorithms F1 scores as well as LROC. Everything must meet a certain threshold greater than 0.8, which is over 80% accuracy. But then it goes through another filter where our scientists, so we have a very multidisciplinary team that's working to both train the algorithms and evaluate the output. So we actually check the relational associations and entity extractions before they go live. And we have a threshold of over 85%.
Okay. Thank you very much. So I have a question for all you together, because we are speaking about AI and personalized medicine. So, and it's linked to the question we have from the public. What are the biggest challenges in using machine learning and data science to improve patient selection and predict treatment response in personalized cancer medicine? So it's more on the technical side, but what are the difficulties or the challenges in using AI in that field? So if someone wants to start, it's too technical, or?
I mean, probably all of us would answer the quality of the data, and having good quality data, which Cassandra said, you have to curate, and that is actually the biggest. I mean, the company was started in 2013. The first product came out, I think, in 2017. By the time that you've curated and managed to get the data in a format in a consistent way, because when you access public systems, the data is there for the user that put it there. It's not there necessarily for anyone else to use as well. So at least in a very general sense, Olivier, I would say, at least from our perspective, it's about getting good quality data and also getting good quality negative and positive data for some of the work that we do, so that everybody publishes the good stuff, right? We've always thought, actually, I've personally always thought it'd be great to have a journal that publishes all the data that all the experiments that didn't work, because that actually would be really valuable as well. And we could maybe start a journal amongst us to do that. But that is actually really important, because then you actually understand what you shouldn't do, because trying to repeat what has worked, as I think Hassan said, from lab to lab, there's variability as well. So it is also really important to have good quality negative and positive data, as well as a volume of data that's well curated for the service, for what we're using it for. I can see a lot of nodding here, so I think I'm probably playing.
And where did you find those quality data? Is it with partnership? Is it open data?
So the open data needs curation, and the more open sources you have, the more curation you need to do. So that's the good quality data can be found when you buy into certain proprietary data spaces, which is what we've done as well. And also working with partners. Large companies tend to have better curated data. So the large farmers that we've worked with have better curated data. And that's probably where it is for us. I don't know about the other guys here on the call.
Okay, so Carola?
Olivier? Oh, sorry, Carola, go ahead.
Yeah.
Sorry, yes, I fully agree. The data that's available for model training is one of the major factors, and getting past the hurdles to get access to certain data sets is certainly as well. One thing we're focusing on a lot currently is also adding to the explainability. So we created this mortality score, but of course down the road, we don't know how our model created this score, but we want to move away from this black box and move maybe more towards a lighter gray to see what does it focus on, how does it generate the score. We've working on kind of creating attention maps to see where is it looking at, where is it pulling information from, how does it put this data together, or looking if the scores that our model generates correlate with known risk factors. For example, in lung cancer, we know age, sex, tumor stage, or TNN. Does this make sense? Or do all of the sudden the stage ones have high mortality scores? Then there is something not right. And this is what potential clients will ask us. Well, how does it get to this point? To just understand what's going on. We're in the process of training another neural network to in isolated anatomical regions, so we can see how much emphasis is put, or how much influence each anatomical region has on the overall score. But just the entire process of explainability, black box, etc., is one of the other major points we've found a bit challenging.
Okay, excellent. Adam, just a second, Alexandra. We'll go to Adam, and then we take the question of the attendees. Okay, Adam.
Sure. I think one of the challenges that we find and is just that framework for responsible AI. I think that's the way I would phrase it, for that ethical implications of AI in what we're doing. And to your point, we're actually a South Wales-based company building a training set of patient blood and data, and is to Vern's point, you need to widen that data set and also look at the framework for how you're using that in an ethical manner, is what I would say.
Okay, thank you. So Alejandro, you have a question from the public, so go ahead.
Yes. This question is from Maximilian Stroby, and actually he's addressing his question to Dr. Schmidt. It says, I'm very much agreed with the notion that better outcome measures beyond RECIST are required. How far does Dr. Schmidt think we are away from seeing digital biomarkers, example based on ML, to getting FDA or equivalent approved as clinical trial endpoints?
Yeah, that's certainly something we looked into as well. We're personally not there. Right now, we're more focused on using our predictions as internal decision-making tools or operational tools like bed planning in hospitals, but very strongly focused on clinical trials, making them official as a biomarker. That's going to be a long way, and they need a lot more data than we could provide currently. So not close, but we're on the way. We're on the way, certainly.
Excellent. Thank you. Hassan, we have 5 minutes. I don't know if you have other questions for...
Yes, so maybe for Carola too. So do you plan to integrate some omics data maybe to your great solution?
Can you repeat that? I was trying to find the question here on the website.
Yeah, do you plan to integrate the using of omics data in your great platform?
we're discussing that currently. We're not quite haven't made a decision, but that's certainly we're keeping in mind. Yes.
Okay, but do you believe that omics data can improve the performance of your platform today?
Yes, I believe that.
Okay.
Thank you.
Alejandro, you have another question from the attendees? Okay, go ahead.
It seems that Dr. Schmidt is having some traction here. Mariam Hani, hi, I'm interested to see that the team of Carola, Schmidt, or other members are working on the prediction outcome of surgery based on 3D data, such as 3D shape as well. Is it part of the projects? If yes, could you please explain it a bit more? If not, just let me know your idea about this kind of deep learning based project. And then parentheses, Mariam is a PhD student working on the prediction of postoperative shape of spine and trying to see similar projects. Thank you.
We haven't thought about this in particular yet, but I'm happy to connect you with our machine learning experts to see if that is something that maybe we can incorporate, or if that would be something we can work on down the road, certainly. But it's not been a major focus for it right now.
Okay, so thank you. Hassan, you have other questions for
last question for Vern. So we know that there is already existing solution for molecule generation. I can give an example. For example, Dassault BioVia, for example, have already existing solution. So how would you place your platform compared to this existing solution, and what's the advantage of your platform compared to this existing solution?
Yeah, and absolutely, and transparently, we're not the only company molecules, design molecules in silico. The difference that we have is the Ligand Express platform, so it's the proteome, is the curated 9,000 to 10,000 proteins that we have, which allows us to actually work on multi-targeted approaches, so targeting more than one particular protein target at any one time when we're designing, but also allowing us to move across the proteome, so in different disease areas as well. That curation and the way in which we identify the potential pockets and the ligand structures, if you like, that would interact with those proteins is something I haven't discussed here, but actually that allows us to work with low data protein structures. So we don't need X-rays, but we do need some 3D protein structure, as well as obviously ligand information as well. So it's different because of Ligand Express. We're the only company that has that as a solution. And we use that to actually both interrogate through phenotypic screening, identifying potential compounds, but also we use that platform to actually help the design of molecules. So using actually the pockets to actually help design the molecules as well. So it's the Ligand Express, frankly, Hassan, that's different, that differentiates us from others.
Excellent.
Thank you so much.
So I think, Alejandro, we have to conclude. So
we have to conclude.
Just before concluding, so just a word for Adam. So sorry, I'm not so familiar with this technology, but I believe that we have a lot of interest on that, and we're gonna have maybe a private session to go more on more details with your solution. So thank you so much for everyone.
Thank you very much, Hassan. That's appreciated.
Okay, so Alejandro, perhaps just before we conclude, can you explain what's going after the creative collision, because there's so much going on after that? So.
Thank you very much, Thomas, Olivier. That's an amazing moderation of the breakout session. Hassan, thank you very much. And everybody else around the table. The discussion was really intense, interesting, and the audience, they were on fire with the conversation. So now follow-ups. As Hassan said, there are some chances that some of you will have a private meeting that we will handle that. We will reach out individually to you and let you know for the next steps. Now for today, we have the pitch competition. Hassan, I think that would be interesting for you too. There's 10 startups that will be competing with a pitch, and they will do so in front of the audience from 3 to 4 something. Later after, at 5, we have networking opportunity. So now for you to know, out of the 600 people that we had like 50 that are following all these conversations, 50% of the 600. Out of those, there are like even numbers between researchers, startups, and companies and other organizations. So I think that networking, I personally have run into precious relationship later after, that someone that I wasn't expected to run into, that brought something very interesting for me later after. So that's the beautiful of tool of networking here in Hopin. We invite you to stay. We are not obliged to, especially if you are in UK or somewhere where it's really late. But otherwise, keep enjoying, and thank you very much for everything.
Yeah. So
Thank you.
Thank you very much. I just followed the numbers on the top of the screen. So we were around 60 to 70 people all along this discussion. So thank you very much for everybody, because we join you really late for some. So you have a short delays to be prepared. So that's really appreciated. I hope that Hassan discover new startups, and they can be a partnership with the Servier and those startups. So thank you very much for that. And perhaps we will see during the networking section at the end. I will be in the Centech booth, so you can join me. And if you want to join the people, they all have their LinkedIn profile, so you can find them on LinkedIn, and then you can just connect with them. So.
Just final comments. I forgot to tell you that for the pitch competition, we will go back to the main stage. To the main stage,
yes. And then for the networking, it's always on your left. So if there's the two hands are crossing, and then you can go there for at 5 o'clock. So thank you very much, everybody.
Just final word. So it's very interesting for me, all those technology, and so I'm very, very interesting to continue discussing with you all. And for sure, we're gonna have a private session with each of you. Thank you very much.
Yeah, we will take care of that.
Thank you.
Thank you.
Thanks for the invitation. Take care. Stay safe, everyone.
Thank you.
Thank you very much.
Bye-bye.
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 Ciao.
Bye-bye.
Thank you.